In this lecture, we'll discuss carbohydrates and glycoconjugates, which are one of the most important biomacromolecules in the living system. First, let's talk about the carbohydrates, which are in many foods you consume pretty much every day. So they play an important role in energy metabolism and storage. And carbohydrates serve as building blocks of polysaccharides, which are the polymers of the monosaccharides, nucleic acids, as well as glycoproteins, and they provide the structural scaffolding for cell walls in plants. And they're involved in molecular recognition that are important in cell growth and transformation. So basically, they have key roles in cell signaling pathways. And they are the most abundant class of organic molecules in nature. And actually, more than half of all organic carbon on Earth is stored in just two carbohydrates, which are starch in plants and cellulose in animals. And the basic formula of carbohydrates are basically repeats of CH2O with the repeats of N, where this N is equals or the larger than 3. And they are also metabolic precursors to virtually all other biomolecules again, about polysaccharides, nucleic acids, glycoproteins, and so on. And the breakdown of the carbohydrates, which is basically the digestion, provides energy that sustains life. So we need to actually consume carbohydrates. Glycolipids and glycoproteins play important roles in structure and recognition too, which we'll discuss later in this lecture and also the next lecture. Let's talk more about the chemical features of carbohydrates. Carbohydrates, especially with at least 3 or 4, depending on its type, 3 or 4 carbons, have chirality, which means that they have one or more chiral centers or the asymmetric centers, and that means they have handedness, so they're going to be a mirrored image too. And these carbohydrates can make the ring structure or the linear structures too. And they form the polymeric structures via glycosidic bonds. And this is not just a linear bonding, but it can also make branches too, with an extensive hydrogen bonding. And the classification of the carbohydrates are the monosaccharides, basically the single unit of the sugar, oligosaccharides, which are few sugars linked together by the glycosidic bonds, as well as polysaccharides, which are the bigger ones with a quite extensive bonding, including the branches too. And there are isomers for the polysaccharides, stereoisomers, epimers, and anomers too. And we're going to discuss all about these different classifications in this lecture. Among different classes of carbohydrates, let's first talk about the monosaccharides, which are the modified polyhydroxyl alcohols. And they typically have 3 to 7 carbon atoms, which are classified based on the number of carbons too. So for example, 3 carbons will form the triose, 4 will form the tetrose, pentose and hexose, and so on. And this is an example of a monosaccharide, which is a D-glucose where it has 6 carbons from 1 to 6 here. And also monosaccharides are classified based on the nature of a carbonyl group. So the monosaccharides that contain the carbonyl group or the aldehyde will be called as aldose, as opposed to the monosaccharides that contain the ketone group will be called as ketose. So aldoses of the 3 or more carbons and the ketoses of the 4 or more carbons have chiral centers. And please remember that the carbons bonded to 4 different groups, all the different groups of those attached to the center carbon will form the chirality, and that chirality causes the rotation of a polarized light. So in glucose, which has the 6 carbons, specifically 4 out of 6 carbons are chiral, which are from the carbon 2 to carbon 5. When you focus on each of these carbons from carbon 2 to 5, you can notice that all different groups, 4 groups are attached around this each carbon, again, which makes the chirality. So it'll have 2 to the 4th equals 16 possible stereoisomers, which have the same constituents but a different atom arrangement. So they're gonna generate the isomers with a different spatial localization of those groups attached to the chiral center. Again, aldoses with at least 3 carbons and ketoses with at least 4 carbons contain chiral centers. And by this point, you should be familiar with the chiral centers as well as chirality and Fischer projection, where these lines actually indicate the different configuration of the different groups. So for example, the horizontal line indicates these groups facing or protruding towards you, as opposed to vertical lines are indicating the groups projecting backward. I mean, not backward, but the back of the paper, so away from you. And also you should be able to tell the L versus D isomers based on those groups, 4 different groups attached to the carbon, the chiral center. So for the monosaccharides, you can use this Fischer projection and can tell the D or L isomers too. And this orientation is decided by the configurations of a hydroxyl group as well as a hydrogen atom at the carbon that is farthest from the carbonyl group. So it's gonna be C5 right here. And this H and OH configuration, or the arrangement, is basically same as this. So it's gonna be the D glucose, as opposed to if we were to have the hydroxyl group here and the hydrogen atom here, that would have been the L glucose. In the same sense, for the monosaccharides with the 2 or more asymmetric carbons, which are the chiral centers, D and L refers to the configuration of the highest numbered asymmetric carbon. Again, highest number of the carbon will be located farthest from the aldehyde. So in case of D-fructose, the highest numbered asymmetric carbon will be this, and the C5 located right here. And since here's an hydrogen atom, and the hydroxyl group is located here, it's going to be the D form. And you can have the mirror image, which is the definition for the enantiomers. We'll have the L fructose, but it really doesn't matter about the arrangement of this C3 or C4 configuration, but it only matters for this C5, which is again has the highest number or the farthest carbon from the aldehyde group, and how these the different groups are attached there. Depending on that, you can tell it as the L form or the D form. And simply you can actually take that as if you can see the hydroxyl group on the right and the hydrogen atom on the left side, it is the D form. When you focus on the carbon that is farthest from the aldehyde. And if opposite, meaning that if OH is here and the H is here, which is this case, that's going to be the L form, L sugar or the L isomer. Those are the same words here. And D form sugars predominate in nature, interestingly, and that's likely due to the stereospecificity of the enzymes that synthesize them, as well as the enzymes that produce this further and for the digestion as well as for energy generation. And for the substances that have multiple chiral centers, just like the carbohydrates or the monosaccharides that we're focusing right now, they can have epimers too. And an epimer is a stereoisomer of another compound that has a different configuration at only one of several stereogenic centers or the chiral centers. For example would be the D-mannose and the D-glucose. Those two are the epimers. That means only configurations at one chiral carbon, which is specifically C2 right here, is different between the D-glucose and D-mannose. All other things, the configuration or the arrangement are exactly the same, but only this C2 are different. So they're epimers. And same is true for the D-glucose and D-galactose too, where the only the configuration at this C4 is different. So this C4 works as a chiral carbon in this case to tell these two are the epimers. But keep in mind that the mannose and the D-galactose are not epimers because obviously there are more than one, actually two of these chiral centers that have different configuration. And this picture shows the structures and different classes of aldoses, which are determined by the number of carbons within the sugars, as well as in their configurations around the chiral centers. So for example, the aldotriose or the triose or the sugars consists of 3 carbons, and glyceraldehyde is an example of that. And aldotetroses are the ones with the 4 carbons. And again, it can have different sugars based on the different configurations. And the aldohexoses contain a lot of the sugars that we are familiar with, such as glucose, mannose, galactose, and so on. And you should be able to tell which is an epimer of which and which molecules are enantiomers. In the similar way, this picture shows the structures and different classes of ketoses, again based on the different number of carbons as well as the configuration of the groups around the chiral centers. And interestingly, the only aldose in nature occurring as monosaccharide is glucose, but several ketoses occur as monosaccharides in nature. But the glucose and other monosaccharides occur as components of larger polymers. So when you look at the polymers such as oligosaccharides as well as polysaccharides, you can actually see a lot of glucose as well as different monosaccharides such as mannose and so on in those polymers. Then how are the polymers such as polysaccharides formed from monosaccharides? The monosaccharides turn into cyclical form to get linked. Then how these cyclical forms are formed in the monosaccharides? They use the most reactive group, which is a carbonyl group. So there's going to be a carbonyl group here as an aldehyde form in aldoses. And also this the carbonyl group exists as a ketone form in the ketoses. And upon their interaction with the alcohol, which contains the hydroxyl group, they're gonna give the hemiacetal or the hemiketal respectively. And this reaction basically happens also within this sugar molecule, which is a linear, but it cyclizes by having an interaction between this aldehyde or the ketone group as well as the hydroxyl group on the opposite end of that linear sugar. So that happens intermolecularly, and we're gonna talk much more in details about this the steps of these reactions in a few slides. And you must be able to draw the structures and their steps too. So as a product, an example would be the pyranoses, which are derivatives of the pyrines, as you can see here, the 6-membered ring. And also furanoses are the common products, which are derivatives of the furans, and they are the 5-membered rings. But you're going to see rarely the 4-membered rings or even 3-membered rings. They exist, but since they have a pretty high energy to contain, they're quite unstable. Now let's take a closer look at the reaction and the steps of the aldose cyclization. And an example that you're seeing here is the pyranose, which is a cyclical form of glucose. And like I told you in the previous slide, alcohols react with the carbonyl groups of aldehydes to form the hemiacetals. So here's the reaction again, especially the OR group in the alcohol attacks the carbon atom within the aldehyde to make the single covalent bond there. So here's the D-glucose, which is a linear form of a sugar, and especially the hydroxyl group here attached to the C5 right here. And the oxygen atom there attached the carbon atom within the aldehyde group in the C1 position to be connected here, make the single covalent bond to cyclize. And it can form this either alpha or beta glucopyranose form. So when you look at it in the different projection here, which is a Fischer projection, you can see that all the other constituents are arranged in the same way except for the C1 position when you look at the hydroxyl group there. So these two are epimers basically. Now this one also shows an example of ketone cyclization by the same reaction as the previous slide. So it uses again the alcohols that react with the carbonyl groups of ketones this time to form the hemiketones. So this shows the reaction here and pretty much the same reaction other than the aldehyde. Now it uses ketones, but also makes the single covalent bond between the carbon and the oxygen atoms from the alcohol to get it cyclizes, especially in the sugar molecules. So starting from the deep fructose, again using the same part here, the hydroxyl group from the C5 and the hydrogen, the oxygen atom attacks the C2 carbon in this case instead of C1, but it attacks the C2 to make the single covalent bond there to form this pentose in this case. But now also it can generate two epimers like that depending on the position of the hydroxyl group. It can have either alpha or the beta fructopyranose. And this is a Fischer projection again that you can see easily how the ring is formed. In the ring form, carbon C1 also becomes chiral. So this is a Fischer projection of the D-glucose in a cyclic form. And as you can see, the carbon 1 actually has the hydroxyl group as well as a hydrogen atom that is attached there instead of the aldehyde in the linear form. So you can give the chirality there, and this is called as anomeric carbon. And anomer is basically the same as epimer, but only happens in the cyclic form. And these anomeric carbons are most reactive. And this alpha, it can have either alpha or beta anomers, and that differs by the configuration of the hydroxyl group at the anomeric carbon, which is a C1. And if the C1, at the C1, the hydroxyl group is located on the right side, then it can be called as alpha configuration in the Fischer projection, as opposed to if the hydroxyl group is in the opposite side, which is the left side of this projection, that will be called as beta configuration. And also in the Howard projection, the alpha and beta can be easily distinguished by the location of this hydroxyl group, again attached to the C1 carbon. If that is located in the same size as this C6 carbon, which is an exocyclic carbon, then it'll be the beta glucopyranose or the beta configuration. But if that is in the hydroxyl group is located in the opposite side from the C6, then it'll be the alpha configuration. And the anomers in different configurations in the alpha and beta interconvert spontaneously via a linear intermediates. And if you take a pure beta form, so 100% of the beta form, with time you will get a mix, which is a racemic mix, which means that there are different stereoisomers that coexist there, would be that mix of 63.6% of beta and 36.4% of alpha. And this phenomenon is called as mutal rotation. And concentration of a linear form at any given point in time is very small. So it's going to be mostly alpha or the beta cyclic form. And we already talked about the Fischer projection, which can show both the linear or the cyclic form of the sugars. And also it can indicate the carbon numbers from C1 to C6, as well as the groups that are attached to the carbon atoms in the right side or the left side, and which can actually indicate the carbonality or the anomers or the epimers too. So we can redraw the same molecules in a different way, which is how about the projection? You should be already familiar with this projection, which shows the carbon, the six-membered rings or the five-membered rings of the sugar. And also the bottom three lines, the thicker lines, indicate that these three atoms or the four atoms are located quite closer to your base, as opposed to the other bondings or the atoms are located quite farther from you. And it can also indicate where the groups are located relatively relative to the plane of the sugar ring. So it's either located on the top or the bottom of that, which can also indicate the carbonality again, anomers or the epimers too. And this anomeric carbon, which is a C1 carbon, is always indicated on the right side, located right side here. And depending on the location of a hydroxyl group, you can tell that if that's the same as the C6, then it will be again the beta configuration, as opposed to the other direction would be the alpha. Cyclic sugars exist as either chair conformations or boat conformations, but they are not going to be in flat conformation due to sp3 hybridization around the carbon atoms within the sugar ring. So the bonds are at 109.5 degrees, and to be a flat one, it should be 180 degrees, which is almost impossible for those carbon atoms that are bonding to each other. And the substituents which are attached to the carbon atoms can be located either equatorially, which is perpendicular to the axis going through the hole in the middle, or axially too, which is parallel to the axis. So in these pictures, the substituents that are labeled as E are basically one that located equatorially, as opposed to the one that ones that are labeled with A will be the one that located axially. And there are a couple of important rules for these sugar rings, the cyclic rings. First one is that bulky substituents, the big one, are more stable in equatorial conformation because it has the least steric crowding. And also the second one is that chair is slightly more stable than the boat. Again, to reduce the steric clashes between any kinds of bulky substituents. The biochemical reactions of monosaccharides are mostly due to the reactions of hydroxyls and anomeric carbons, which are the most reactive groups in the sugar molecules. And the carbonyl, which is an anomeric carbon, can react with alcohols to produce acetyls and ketols. And monosaccharides can form the glycosidic bonds too, which is a bond connecting anomeric carbon to acetyl oxygen. This picture shows the example of forming the glycosidic bond. So the D-glucose reacts with the methanol in addition with the hydrogen cation to form this glycosidic bond. It can happen in either both alpha and beta conformations too, and especially it attacks or connects this anomeric carbon, which is a C1 carbon, and the acetyl oxygen here as an OCH3. And one of the most important glycosidic bonds are the N-glycosidic bonds that we have already seen when we are studying about the nucleotides. Here the DNA molecule is shown, and as you can see, it uses the pentose, the sugar molecule too, and it is connected to the other the base, nitrogenous base, basically through this nitrogen atom. So that's why it's called N-glycosidic bond. So the connection to the sugar molecule. Oligosaccharides, which are dimers and trimers of the monosaccharides, as well as polysaccharides, are formed by making glycosidic bonds between the monomers and the monosaccharides. And there are various types of oligosaccharides as well as polysaccharides, such as lactose, maltose, sucrose, and so on. And there are several factors to determine the type of these oligosaccharides. The first one is, of course, the components, the monomers that are forming these oligosaccharides. So it can be galactose, glucose, fructose, and so on. And also their linkage type determines the identity of the oligosaccharides. So it can form the 1,4 linkage, 1,2, or even 1,6,2. And there are a lot of oligosaccharides that are used daily. So the lactose is commonly found in dairy products, and sucrose is a common table sugar. Again, the name of oligosaccharides or the polysaccharides are based on the names of the monosaccharides that are involved there, as well as the linkage too. So there are two examples here. The first one is sucrose, which has the glucose and fructose. So it'll be called as glucose fructose, but also indicates the conformation. Here, as you can see, the hydroxyl group at the anomeric center, or the anomeric carbon, is located on the opposite position from the C6 carbon. So it's gonna be alpha configuration, and the glycosidic bond is forming to the C2 carbon here. So it's gonna be 1,2 fructose. So the full name will be glucose alpha 1,2 fructose. Another example is the lactose. Here again, it has the galactose as well as glucose. And also when you look at the galactose, now the hydroxyl group, which should be located on here on the above of the C6 ring here, which is on the same position as the C6 ring, so it'll be beta configuration. And this C1 carbon is connected through the glycosidic bond to the C4 of the glucose. So it's gonna be galactose beta 1,4 glucose. Oligosaccharides as well as polysaccharides have reducing end and non-reducing end. And non-reducing end is basically the anomeric positions of the sugar located in the middle of these polymers. So this anomeric position or the carbon is already consumed or used for making this glycosidic bond. So there's no possibility or the potential that this sugar is opened up to get the linearized form to have the aldehyde or the ketone group recovered there, as opposed to the sugar located at the end of this oligosaccharide now have the reducing end where this ring can be open and basically restores this carbonyl group, which is a part of the aldehyde or the ketone, and especially at the anomeric carbons. And this property of reducing end and non-reducing end can be used to test that if any sugars that you have are the monosaccharides mostly, or the oligosaccharides.